Okay, so we're going to review DNA, junk DNA, transcription, and a little bit about control of transcription. So if this section of DNA is our gene, it's important to remember or know that not all sections of this DNA are actually going to be useful with respect to coding for a protein. So the quote-unquote useful parts we call exons. And these exons are basically the sections of DNA that will code for a protein. Now, what that means is that there are actually sections of DNA that are not useful and do not code for DNA, and these are called introns. Now, they're not completely useless. Using the term junk DNA is a little bit incorrect because they're not junk, they are some useful parts to this. And most of those useful parts are for controlling whether or not we transcribe this gene. So one part of this intron will be the promoter. And this is basically our start line. This is where our RNA polymerase will start. And similarly, at the end, we have something called the terminator, which is going to indicate the place to stop. And so what this means is, and I'll draw a very quick diagram here, is that our DNA looks something a little bit like this at the point that it's going to be transcribed. So I'm just going to write P for promoter and T for terminator. Okay, is that our enzyme, RNA polymerase, is going to start at the promoter and move its way along and as it's done this, moving in that kind of 5', 3' direction, is going to be creating mRNA. And that mRNA is going to be a copy of the antisense strand, which has acted as a template. The opposite strand being the sense strand. And what that means is that across that process of transcribing, we will have taken in the section that is both coding and non-coding. So our mRNA that we will end up with will have all of those sections copied. So we can already see that our introns are important in terms of regulating where to start and where to stop transcription. Okay? So we'll get a little bit more into control of that in a second. What I just want to touch on now is post-transcription modification. And it basically does what it says on the tin. That after transcription, we're going to modify that piece of mRNA. So here is our pre-mRNA, which has obviously got both our coding and non-coding sections in. And actually all that we need is the coding sections. So what happens is that we end up with the formation of a complex which is known as a spliceosome. So a complex of proteins are going to come together and they're basically going to fold that mRNA in on itself so that the intron, the junk DNA, the part that is not going to code for any protein, is actually going to be taken out. And so that next step of the process means that we end up with a loop of mRNA, which is basically useless because this is the intron. And now we will also have a stretch of mRNA, that will only contain the coding sections that were taken from the DNA. And this is now, or it will be soon, known as mature DNA, mature mRNA. Now, the only other things to happen at this stage is that we will add a 5' cap, or a modified 5' cap. It's going to be a modified guanine. And we also add a poly A tail. And all those two things are going to do is to try and help reduce degradation after it's going through a ribosome as part of translation. So bits will break off, but we're not going to damage the mRNA itself, and it can get used more than once. Now, the other interesting part of this is that if we have our pre-mRNA and we actually see that we have a few more introns than normal, so let's say we have 1, 2, 3, 4. We can also go through this process called alternative splicing. And what this means is that one gene can produce multiple different proteins, because different proteins can be produced from splicing different sections of DNA. So, for example, we might actually have a stretch of DNA that just includes the first exon, the second exon, and in this case, maybe the fourth. And that this will make protein A. We might have a situation whereby we actually have 1, 3, and 4 together. And this will make another protein, protein B. So all we're doing is saying that one gene can actually produce multiple different proteins based on different splicing of those genes. And we'll leave it at that, and I'll come back and talk to you about enhancers and silencers.